You know, there's this really specific assumption we make when we go for a walk in the woods.
Oh, absolutely. Like we just see it as a backdrop.
Right. You step onto a trail, you look around, and you assume you are looking at this passive landscape, just a quiet, tranquil painting. You step over a fallen log, you see a plain, creamy white mushroom sitting there on the damp earth, and you just register it as background scenery.
Yeah, it's a very common human bias, I mean, to
Yeah.
view flora, and especially fungi, as these quiet, unassuming little background characters. We just think they're waiting around for animals to interact with them. We assume they are totally at the mercy of their environment.
But the reality is like completely different. If you stop and actually look closer at this one specific creamy white mushroom, that entire tranquil landscape painting is just ripped to shreds.
It really is.
Because you are not looking at a passive piece of scenery. You are looking at a master of deception, an organism that is actively engaged in this invisible, ancient, and honestly highly aggressive biological arms race.
The word aggressive is doing a lot of heavy lifting there,
Right.
but it is entirely justified. What we are discussing today is basically the absolute definition of a biological wolf in sheep's clothing.
Which brings us to our mission on this deep dive. We have gathered a stack of incredibly diverse sources for you today. I mean, we are pulling from deep-cut taxonomic phylogenies, heavy metal biogeochemical studies, 16th century Polish herbals, and even some really cutting-edge 2024 to 2026 neuropharmacological research.
It's a wildly diverse stack of papers.
It is, and what is so striking is that all of these diverse, seemingly unconnected fields of study all point back to one organism. Lactifluus piperatus, commonly known as the peppery milkcap mushroom.
And it serves as just the perfect subject for our analysis today because its physical appearance is so remarkably simple. But underneath that unassuming creamy white cap, the sources reveal an organism that is secretly, well, a biochemical warfare factory.
Oh, totally.
It operates as a master forest engineer dictating the growth of giant trees. It functions as a heavy metal biofilter that can endure toxic industrial waste. And, as the most recent pharmacological literature suggests, it might literally hold the blueprint for the future of synthetic pain medication.
It's just mind-blowing. So I want to start by untangling the massive identity crisis this organism has been going through because it sets the stage for that deception we just talked about.
Right, the taxonomy issue.
Yeah, for hundreds of years, the scientific community thought they had this mushroom completely figured out. But the 2024 to 2026 research proves that the foundational texts of mycology were essentially lying to us.
Well, lying implies intent, you know? But our classification system certainly failed us. To understand the magnitude of this failure, we really have to look back at the origins of modern biological naming.
Back to Linnaeus.
Exactly. In 1753, Carl Linnaeus, the architect of taxonomy himself, officially described this mushroom. He categorized it based purely on human observation, right, what he could see. And he named it Agaricus piperatus.
From the Latin.
Right, the Latin root piperatus means peppery, which was a direct reference to the mushroom's legendarily intense burning taste when it gets broken.
A burning taste that we will definitely be exploring in a minute because peppery does not even begin to cover the actual physical trauma this thing inflicts.
Not even close. But Linnaeus' classification stood for a very long time. This mushroom actually became the poster child for a massive category of fungi.
It became the anchor, right?
It did. For centuries, this specific mushroom served as the type species for an enormous genus called Lactarius. Now, in taxonomy, the type species is the gold standard. It is the physical reference point for everything else.
That's the defining example.
Exactly. And the defining morphological feature of the Lactarius genus is that when their tissues are damaged, like if you cut the cap or break the gills, they exude this milky, opaque latex.
That is why they are literally called milk caps.
Right. So if a botanist in the 19th or 20th century wanted to understand what a true milk cap was, they were told to go look at the peppery milkcap. It was the absolute template for the entire group.
So the logic was basically, it looks like a milk cap, it bleeds milk like a milk cap, therefore it is the ultimate milk cap.
Which makes perfect sense if you were just relying on human eyeballs.
Right. But then we hit the molecular phylogenetics revolution between, 2008 and 2011.
Yeah, right around there.
And science stopped looking at just the shape and the milk and started reading the actual underlying source code, the DNA. And that is when the entire Lactarius family tree just collapsed.
It was a seismic shift in mycology. When researchers began sequencing the DNA, they discovered that the Lactarius genus was what biologists call polyphyletic.
Let's break that term down for the listener. Polyphyletic means they don't share a single, exclusive, common ancestor, right?
Correct. Imagine finding a group of people who all have bright red hair and just assuming they must all be part of the same immediate family.
Makes sense on the surface.
But then when you run a genetic test, you find out one person is from Ireland, one is from a completely unrelated lineage in Asia, and another is from South America. They developed the same physical trait entirely independently. That is polyphily. The lactarius grouping was based entirely on a superficial trait, the ability to exude latex.
So the milk was basically just a biological coincidence, not a shared family heirloom.
Precisely the issue. So the taxonomic hierarchy had to be aggressively reorganized. And the most shocking casualty of that reorganization was the founding father itself.
The Peppery Milkcap.
Yep. Lactifluus piperatus was ejected from the Lactarius genus entirely. It was moved into a newly defined genus called Lactifluus and nestled into a highly specific subgenus also called Lactifluus within the section Pepperati.
If I can try an analogy here, because I love analogies, that is like historians suddenly discovering that George Washington wasn't actually an American colonist.
Okay, let's hear it.
Like, he was actually a deeply entrenched foreign operative from a completely unrelated nation who just happened to be standing in the room when the Declaration of Independence was signed. The guy we based the whole system on didn't belong to the system.
That actually captures the shock among mycologists perfectly. The organism they used to define the genus for hundreds of years didn't even belong in it.
But the deception doesn't stop there. Because the sources tracking the latest DNA sequencing from 2024 to 2026 reveal something even more profound.
Yes, the cryptic complex.
Exactly. We didn't just misclassify the genus, we completely misunderstood the species itself. We used to think that the peppery milkcap was just one single globally distributed mushroom.
For decades, the assumption was that if you walked into a beech forest in Europe and found a peppery milkcap, and then flew to a deciduous forest in North America and found one there, you were looking at two members of the exact same species.
Because they look exactly the same.
To the naked eye, under a microscope, and even chemically, they appear identical. But modern genomic data shattered that assumption.
The papers specifically point to sequences like the internal transcribed spacer, the ITS, along with the large subunit 20ness RNA and RPB2 sequences. What exactly are those sequences doing that proves these mushrooms are, lying to us?
To understand why those specific sequences are used, we really have to look at how cells manufacture proteins. Every living organism uses ribosomes, these cellular machines that build proteins.
Right.
The DNA instructions for building these ribosomes are incredibly ancient and essential for survival. If a random mutation fundamentally changes a ribosomal gene, the organism usually just dies.
So they don't change very often.
Exactly. They change very, very slowly over millions of years. They are highly conserved. However, between those highly conserved ribosomal genes, there are stretches of DNA called the internal transcribed spacer, or the ITS.
And what does the ITS do?
Well, these sections don't actually code for functional proteins. Because they aren't critical for immediate survival, mutations can accumulate in the ITS regions freely without killing the fungus.
Oh, I see.
This makes the ITS a perfect evolutionary tape recorder. By counting the accumulated mutations in the ITS, scientists can track exactly how much time has passed since two organisms shared a common ancestor.
So it's a molecular clock.
Precisely. So when they checked the tape recorder on the European peppery milkcap and compared it to the North American one,
The clocks didn't match.
not even close. The data proved that the European populations are entirely genetically isolated from the North American ones. They do not interbreed. They are completely distinct genetic lineages that simply evolved to look identical.
A cryptic species complex.
Exactly. And once researchers realized they were dealing with a cryptic complex, they started finding hidden lineages everywhere.
Like the new one highlighted in the 2026 data, found in a completely unexpected environment.
Yes, mycologists recently identified Lactifluus luteopallitus, a brand new member of this cryptic complex. And they didn't find it in a temperate European woodland.
Where was it?
They found it thriving in the tropical oak forests of eastern Mexico.
Wow. My analogy for this cryptic complex is like discovering that your ordinary, completely boring neighbor John isn't just one guy.
Okay, another analogy.
Stick with me. He's actually a network of genetically distinct, identical-looking clones operating globally under the same alias. You think you know John, but European John and tropical Mexican John are entirely different operatives running the same software.
It's an apt comparison.
And honestly, this makes me deeply skeptical of taxonomy in general. I mean, if morphological classification, just looking at things with our eyes and microscopes, was this catastrophically wrong about such a famous foundational mushroom, how many other cornerstone species in biology are secretly a dozen different organisms hiding in a trench coat?
It really forces a necessary humility upon the scientific community. For centuries, our entire understanding of the natural world was constrained by human perception.
We just trusted our eyes.
We operated under the assumption that physical similarity equaled genetic relation. The Peppery Milkcap complex proves that nature is vastly more complex, layered, and deceptive than our visual classification systems could ever account for.
And that theme of hidden identities, of a calm exterior masking a radically different interior reality, sets up the most staggering part of the literature we reviewed today.
The chemical defense.
Yes, because the peppery milkcap isn't just hiding its true genetic lineage in the soil, it is literally stockpiling a latent, hyper-aggressive chemical weapon system that only deploys the second it is attacked.
And calling it a chemical weapon system is entirely scientifically accurate. The biochemical sophistication of this defense mechanism rivals engineered munitions.
Okay, so let's break down this biochemical warfare because the mechanics of it are unbelievable.
Yeah.
If I am walking through the woods and I see an intact peppery milkcap sitting quietly on the soil and I were able to magically analyze its internal tissues without causing any physical damage to it whatsoever, what would I find? Is it toxic just sitting there?
You would find a completely harmless organism. An uninjured peppery milkcap contains chemical compounds that are biologically inactive and completely tasteless.
What? Completely tasteless.
Yes. Specifically, the fungus synthesizes fatty acid esters of sesquiterpene alcohols. The primary compound in the resting state is called steroylvelutinol.
Steroylvelutinol. So that is sitting inside the mushroom cells.
Correct. The fungus expends a massive amount of metabolic energy to build this complex molecule, and then it simply sequesters it away inside specialized cellular compartments.
Just putting it in storage.
As long as the structural integrity of the mushroom is maintained, these chemicals are inert. They do absolutely nothing. They are essentially a stockpiled arsenal locked behind a biological blast door.
But the millisecond that blast door is breached, say I step on the mushroom, or a wild boar steps on it, or, a tiny little forest slug takes one microscopic bite out of the gills, what happens inside the cells?
The physical damage ruptures those specialized cellular compartments. The blast doors are blown off. This allows the inert steroylvelutinol to flood out and mix with specific enzymes, esterases, and lipases that were stored in completely separate areas of the tissue.
So the weapon and the detonator are kept in different rooms until the wall between them collapses.
A perfect way to visualize it. And the moment they mix, it triggers a high-speed enzymatic cascade that happens in a matter of seconds.
Seconds. That is incredibly fast.
It has to be. First, the enzymes hydrolyze the ester group, meaning they use water to cleave the stearic acid off the molecule. What is left behind is a highly volatile structure containing an oxirane ring.
Wait, hold on. An oxirane ring. Walk us through why that matters. What is the geometry they're actually doing?
Right, so in organic chemistry, an oxirane ring, which is also known as an epoxide, is a three-membered ring consisting of one oxygen atom and two carbon atoms.
A triangle.
Yes, because it is a triangle, the chemical bonds are forced into incredibly tight, unnatural angles. Imagine taking a thick piece of flexible plastic and bending it into a tight triangle.
It takes force to hold it there.
Exactly, and it desperately wants to spring open. The oxirane ring is under massive structural tension.
So the enzyme basically pulls the pin, and the spring-loaded ring violently snaps open.
Yes. The ring opening releases that tension, driving a rapid rearrangement of the underlying chemical framework, which is called a Mera's main skeleton.
Oh, wow.
The molecule practically folds in on itself, converting into entirely new shapes like lactarane and cyclolactarane forms.
Wait, I need to synthesize this for a second. It is a biological binary weapon.
Very much so.
You have two completely harmless, inert components stored safely apart, but the absolute millisecond the hull of the mushroom is compromised, they mix, the structural tension snaps, and they instantly synthesize into a live munition.
It is instantaneous chemical manufacturing on the cellular level. The end products of this violent rearrangement are compounds known as 1,4 dialdehydes.
And the literature identifies the primary active weapons as velleral, isovelleral, and piperadiol, right?
That's correct.
And what do these dialdehydes actually do? Like, why did the mushroom go through all this trouble to make them?
They transform the previously harmless, creamy white latex of the mushroom into a highly pungent, mutagenic, and cytotoxic fluid.
Cytotoxic meaning it destroys living cells.
And mutagenic meaning it actually damages DNA. This is an uncompromising anti-feeding strategy. The fungus is actively trying to inflict tissue damage on the mouthparts of whatever animal just bit it, effectively teaching that predator to never, ever make that mistake again.
The literature describing the human experience of this defense mechanism is honestly intense. I mean, we absolutely must state for the listener, do not try to taste this mushroom in the wild.
Emphatically do not.
But mycologists who have sampled a microscopic drop of the latex for identification purposes, they do not describe it as spicy, like a salsa.
No, not at all.
They describe a searing, metallic, face-melting heat that lingers for an agonizing amount of time. And the neuropharmacology behind why it feels like that is a masterclass in evolutionary warfare.
It is. To appreciate the nuance of this chemical weapon, we need to compare it to a biological burning sensation that most people are actually familiar with.
Like chili peppers.
Exactly. If you eat a jalapeno or a habanero pepper, the active chemical causing the heat is capsaicin. In mammalian nervous systems, capsaicin specifically binds to a pain receptor called the TRPV1 channel.
TRPV1, so that's the chili pepper receptor.
Essentially, yes. TRPV1 is designed to detect actual thermal heat like touching a hot stove to warn your brain of physical burning. Capsaicin basically tricks the receptor into firing, making you feel heat that isn't physically there.
Right.
Now, one might assume the serine dialdehydes in the Peppery Milkcap also target TRPV1, but they don't.
This is the crazy part.
The compounds like Isovelleral bypass the chili pepper receptor entirely and hunt down a much more severe target, the TRPA1 channel.
TRPA1, I want you, the listener, to really picture this. Think about the last time you chopped jalapenos, washed your hands, and then, accidentally rubbed your eye an hour later. You felt that sharp, hot TRPV1 sting. Now scale that up to TRPA1. What is this receptor actually meant to detect?
The TRPA1 channel is a nociceptive receptor, a profound pain sensor, that evolved to detect severe environmental irritants and signal inflammatory pain.
Like what kind of irritants?
It is the exact same receptor that is triggered by tear gas, mustard gas, and the crushing intensity of raw wasabi or horseradish.
Wow.
It doesn't signal warmth. It signals deep, tissue-damaging chemical trauma.
So it's literally the tear gas receptor, but it is the way the mushroom attacks this receptor that is so brutal.
The mechanism of action is terrifyingly efficient. Normally, when a chemical messenger interacts with a receptor, it is a transient connection.
It bounces off.
Right. The molecule bumps into the receptor, the pain signal sent to the brain, and then the molecule floats away or is broken down. It is temporary. But isovelleral forms a covalent bond with the cysteine residues located on the TRPA1 channel.
Okay, I want to make sure the listener understands the gravity of a covalent bond here. If a normal chemical interaction is like a high five, you touch, the signal goes through, you pull your hands apart, what is a covalent bond?
A covalent bond means the two molecules are now sharing electrons. They are physically fused. To use your analogy, it isn't a high five. It is coating your hand in industrial superglue, slapping the receptor and refusing to let go.
Oh my God, so the pain receptor is locked in the on position?
Yes. Because the molecule is covalently bonded, the TRPA1 channel cannot easily reset. It continuously floods the nervous system with agonizing signals of inflammatory chemical damage.
Just non-stop pain.
Exactly. It takes a significant amount of time and cellular effort for the body to clear that bond. But the evolutionary brilliance of this system contains one final, almost unbelievable twist.
The receptor blocking. This blew my mind when I read it in the pharmacology papers. Explain what isovelleral is doing to the chili pepper receptor while it is superglued to the tear gas receptor.
While isovelleral is aggressively triggering the TRPA1 channel, it simultaneously acts as a profound antagonist to the TRPV1 channel.
Wait, antagonist meaning?
Meaning it chemically blocks it. It turns the chili pepper receptor off. It prevents TRPV1 from firing at all.
So let me wrap my head around this. You have a mushroom that inflicts a chemical burn far hotter and more agonizing than the hottest chili pepper on Earth, but it actively secretes a compound that shuts down the body's ability to feel chili pepper heat.
Precisely.
It is literally scrambling the predator's sensory dashboard. Why would it evolve to do that? What is the evolutionary logic of turning one alarm off while smashing the other one with a hammer?
This is where we look at the evolutionary arms race between fungi and mycophagous or fungus-eating animals.
Okay.
Over millions of years, it is highly probable that ancient rodents or forest slugs began to develop a tolerance to TRPV1 activation. They essentially built up a resistance to capsaicin-like compounds. So the mushroom's defense was becoming obsolete.
Oh, the rodents started liking the spicy food?
In a manner of speaking, yes. So the peppery milkcap bypassed the obsolete alarm system entirely. It evolved a compound that actively suppresses the compromised receptor, TRPV1, and instead violently targets the TRPA1 chemical damage receptor, ensuring the predator still receives a paralyzing pain signal that it cannot adapt to.
It is an evolutionary countermeasure of the highest order.
It really is.
And what would a TRPA1 chemical assault actually look like for a human? Say a forager ignores the warnings, gets this milky latex on their fingers, and casually wipes sweat out of their eye.
It would be a medical emergency. Because TRPA1 is fundamentally linked to inflammatory pathways, the eye wouldn't just feel warm. The blood vessels would engorge. The tissue would become massively inflamed, mimicking a caustic chemical burn.
That sounds horrific.
The eye would water profusely in a desperate attempt to flush the covalent bonds, which wouldn't work easily. The sustained pain would be debilitating. This is exactly why expert mycologists handle this specific species with immense caution, often wearing gloves when sectioning it for study.
It is an utterly uncompromising hostile defense mechanism, and reading about this binary tear gas weapon, it paints the peppery milkcap as this isolated, aggressive entity that views the entire forest as an enemy.
Which is only half the story.
Right, because when we transition away from the cap and look at the research detailing what this organism does beneath the soil, the narrative completely inverts. It doesn't just look out for itself. Beneath the dirt, it utilizes its immense biochemical power to act as the ultimate caretaker of the forest.
The contradiction is fascinating. The hostile chemical factory above ground is actually one of the most vital collaborative partners below ground. The peppery milkcap is what ecologists classify as an obligate ectomycorrhizal fungus, often abbreviated as ECM.
The word obligate being the key there, it means it cannot live alone.
Absolutely. Lactifluus piperatus cannot simply digest dead leaves and survive. It is obligated by its genetics to seek out and form a symbiotic partnership with the living root systems of specific canopy trees.
What kind of trees are we talking about?
The northern hemisphere, it primarily seeks out beech, hazel, and oak trees.
And this symbiosis isn't just like a gentle handshake in the soil. It is a profound structural integration. The fungal threads, the mycelium, actually wrap around the microscopic root tips of the tree like a tight biological glove.
A structure known as the Harding net. The traditional textbook view of this relationship is relatively straightforward. The massive tree performs photosynthesis, generating vast amounts of complex sugars from sunlight and carbon dioxide.
And the fungus wants that sugar.
Exactly. The fungus, which lives in the dark and cannot photosynthesize, needs those sugars. In exchange, the fungus acts as a highly efficient, microscopic extension of the tree's root system, mining the soil for water, phosphorus, and nitrogen, and trading them to the tree for sugar.
A simple barter system, water for sugar. But our deep dive sources show that the peppery milkcap refuses to just be a passive trading partner. It doesn't wait for the tree to grow roots. It actively forces the tree to grow them.
The literature refers to this behavior as the forest mother concept.
Yes, I love this.
The fungus operates as an external master engineer of the plant's development. It achieves this by synthesizing and actively exuding plant hormones called auxins directly into the surrounding soil. The primary auxin it manufactures is indole-3 acetic acid, or IAA.
So the mushroom is brewing the exact same rooting hormones that you would buy at a garden center to clone a plant, and it is pumping them into the root zone of wild oak seedlings.
Exactly. It is a highly orchestrated manipulation of the host plant's cellular machinery. And the sources break down this fungal-induced root development into 4 distinct, chemically driven stages.
Let's walk through those stages S0 through S3, because the level of control the fungus exerts here is astounding. It starts with S0, the pre-emergent stage.
Right, so S0 occurs before any new root structures are even visible. The fungus detects the tree root and begins flooding the area with IAA. This hormone surge penetrates the plant's tissues and triggers a massive synthesis of antioxidant enzymes within the plant cell.
Wait, why antioxidants? What is the fungus protecting the tree from?
From the stress of rapid growth. When cells divide at an accelerated rate, they generate dangerous byproducts called reactive oxygen species, free radicals that can tear apart cell walls and DNA.
The metabolic exhaust.
Precisely. By forcing the plant to build a stockpile of antioxidants first, the fungus is essentially prepping the biological soil. It is ensuring that when the rapid growth phase begins, the new cells won't be immediately destroyed by their own activity.
That is an incredible level of foresight for a fungus. So the blast shielding is in place. What happens in S1?
S where is early formation. The fungal auxins override the plant's normal growth inhibitors and trigger rapid mitosis cell division in the pericycle layer of the root. A massive cluster of new cells begins to bulge beneath the surface.
Which leads directly to S2, the massive formation stage. This is where the root bursts out. But the literature uses a very specific term here. It says the fungus forces the creation of a nutrient sink. Explain what a sink is in this context.
In plant biology, a sink is an area of the plant that demands and consumes huge amounts of resources, like a growing fruit, or in this case, a massive cluster of new lateral roots. By forcing the tree to develop this dense, tangled mass of new roots, the fungus is effectively forcing the tree to pump huge amounts of its photosynthetic sugar down into this specific area to fuel the growth.
So the fungus isn't just being a kind mother. It is strategically building a massive feeding trough for itself and forcing the tree to fill it with sugar.
It is a mutually beneficial manipulation, but a manipulation nonetheless.
Right.
Finally, in stage S3, late formation, the root structure is fully emerged. The fungus continues to supply hormones to maintain the root meristems, the actively growing tips, and encourages complex, multidirectional branching.
Perfectly shaping the roots to maximize the surface area for the fungal mycelium to attach to.
Exactly.
It hacks the tree's development to build a custom-designed home for itself. But the caretaking, or the manipulation, depending on how you look at it, doesn't stop at just pumping auxins. We have to talk about the soil ecology, specifically what this fungus does when it encounters industrial pollution, because the heavy metal biofilter data from the 2025 papers is astonishing.
This is arguably one of its most vital ecological roles in the modern era. Lactifluus piperatus has been identified as an extreme hyperaccumulator of heavy metals, with a specific affinity for lead.
To give you, the listener, some context on lead levels, if you go out to a pristine, ancient woodland or even just a normal, healthy garden, what is the background level of lead in the soil?
In healthy, unpolluted surface soil, the global mean for lead concentrations usually falls between 10 and 67 milligrams per kilogram of soil. That is the baseline the ecosystem evolved to handle.
But the studies look at industrial areas, old manufacturing plants, heavy mining sites, places where the soil is just deeply contaminated. What kind of numbers are we seeing there?
In heavily polluted anthropogenic sites, the lead levels can skyrocket to catastrophic concentrations, frequently exceeding 10,000 milligrams per kilogram.
10,000? That's insane. And what does 10,000 milligrams of lead actually do to a tree seedling trying to grow in that soil?
It is lethal. Lead is highly toxic to plants because it mimics essential minerals like calcium and zinc.
So the plant gets confused.
Right. The plant roots absorb the lead, thinking it's a nutrient. But once inside the cells, the lead binds to critical enzymes and fundamentally disrupts them. It halts photosynthesis, shatters DNA, and suffocates cellular respiration. A young oak tree, attempting to sprout in that environment, will simply wither and die.
Unless it is partnered with our peppery milkcap.
Precisely. If the tree has formed an ectomycorrhizal symbiosis with Lactifluus piperatus, its survival odds increase dramatically. The fungal mycelial network acts as a vast, incredibly efficient biological sponge stretching out into the contaminated soil.
But wait, how does the fungus not die from the lead?
Fungi have evolved vastly superior detoxification mechanisms compared to plants. When the peppery milkcap absorbs the lead from the soil, it doesn't let the metal roam free in its cells. It actively pumps the toxic lead ions into specialized, heavily shielded cellular compartments called vacuoles and vesicles.
Oh, wow. It locks the lead in a biological safe.
Exactly. It sequesters the heavy metal, crystallizing it or binding it to specific proteins so it remains immobilized. The massive consequence of this is that the lead is trapped within the fungal tissue and never passes through the Harding net into the host tree's delicate root system.
That's incredible.
The fungus essentially filters the poison out of the environment, allowing the oak tree to thrive in soil that should by all rights kill it.
Okay, I have to challenge the evolutionary intent here, because this sounds incredibly noble, right? The selfless fungal guardian taking a toxic bullet for the majestic oak tree.
That sounds nice.
But evolution rarely operates on nobility. We know the fungus needs the tree alive to get sugar, but we also discuss the concept of elemental defense in the literature. Is it possible the mushroom is hoarding this toxic lead for a much more selfish, aggressive reason? Like, is it trying to poison its own predators?
That is a highly debated, incredibly compelling hypothesis in modern ecology. Let's look at the mechanics. By hyperaccumulating massive concentrations of lead in its own tissue, the fungal mycelium itself becomes acutely toxic.
Right.
Now, imagine a soil-dwelling nematode, a root-eating insect, or a grazing microarthropod that attempts to feed on this massive, sugar-rich fungal network.
They take a bite, and instead of just getting fungus, they get a lethal, concentrated dose of heavy metal poisoning?
Exactly. The elemental defense hypothesis suggests that hyperaccumulation is a brilliant dual-purpose strategy.
It's a win-win for the fungus.
Yes. The fungus cleans the immediate environment to keep its sugar supplier alive, while simultaneously utilizing the ambient industrial pollution to turn its own body into a toxic, unappetizing minefield for predators.
A selfish landlord deliberately hoarding toxic waste in the walls to kill any pests trying to chew through the drywall. It is ruthless, and it is genius. But this brings us to a massive, glaring contradiction that we really have to unpack.
The human element.
Yeah. We have just spent the last half hour establishing two absolute facts about this organism. One, it acts as a literal sponge for industrial lead. Two, it manufactures a binary chemical weapon that unleashes face-melting, tissue-damaging dialdehydes the second you break its skin.
Yes. Both highly toxic attributes.
And yet, when we turn to the historical texts we've gathered, it defies all modern logic and self-preservation that humans have been actively hunting, harvesting, eating, and prescribing this incredibly dangerous organism as medicine for hundreds of years. The historical record shows people deliberately seeking this out.
It seems paradoxical, but it actually highlights the immense ingenuity of historical pharmacology. When we examine the records from the 16th and 17th centuries, particularly focusing on Eastern Europe, we find incredibly detailed documentation of this mushroom's integration into medical practice.
Our deep dive sources specifically highlight the foundational work of Polish herbalists and physicians.
The texts point to figures like Stefan Folomers, Marcin Cienek, and Simon Serenius.
These weren't just like village healers.
No, these were not folk healers operating in a vacuum. These men were legendary figures in the history of European botany and medicine. Their comprehensive herbals, massive, painstakingly illustrated encyclopedias of local flora and their medical applications, were the medical standard of their era.
And many of these original texts are currently preserved in the archives of the Ossoliński National Institute in Poland, right?
Correct.
And when you open those ancient Polish texts, they don't say avoid the burning milk mushroom. They prescribe it for very specific, very painful ailments. The sources say they used it primarily as a diuretic and as a lithotriptic.
They did.
Okay, most people know a diuretic makes you urinate, but what exactly is a lithotriptic?
In historical and modern medicine, a lithotriptic is an agent specifically utilized to dissolve, crush, or facilitate the expulsion of kidney stones and renal calculi from the urinary tract.
Okay, I'm going to push back incredibly hard here on behalf of the listener. We just established this organism hyperaccumulates lead and makes blistering chemical weapons. I do not want anyone listening to this deep dive to think, oh, I have a kidney stone, I'm going to go to the industrial park behind my house, pick a Peppery Milkcap and eat it. Shouldn't the takeaway here be that 16th century doctors were just wildly guessing and poisoning their patients?
Let's pause and separate the modern context from the historical one, because the distinction is vital here.
Okay, fair.
First, regarding the lead hyperaccumulation. Heavy metals are only present in the mushroom if they are present in the soil. A peppery milkcap harvested by a 16th century Polish doctor in a pristine, pre-industrial ancient woodland would contain absolutely zero industrial lead. It is not inherently toxic with heavy metals, it simply reflects its environment.
Okay, fair point. Pre-industrial revolution forests didn't have battery runoff. But what about the organic chemical weapons? The velleral and isovelleral? They still had the tear gas mechanism back then. If you eat it raw, the biological airbag deploys in your mouth.
And that is exactly why the historical texts never prescribed eating the mushroom raw. These practitioners were highly observant. They utilized specific multi-stage preparations, drying, boiling, and rendering the mushroom into powders and tinctures.
Which destroyed the enzymes that trigger the oxirane ring.
Precisely. They empirically figured out how to disarm the chemical weapon. And what is truly fascinating is that modern biochemical analysis has validated their use of the mushroom for kidney stones. Those ancient doctors were actually onto something biochemically real.
Walk us through the modern validation. How does a disabled forest mushroom actually cure a kidney stone?
It comes down to the mushroom's extreme mineral density. When modern food scientists analyze the nutritional profile of Lactifluus piperatus, they find it has an exceptionally high ash content, averaging around 5.38% of its total dry weight.
Ash content being a nutritional term, not literal fire ash.
Correct. In nutritional analysis, ash content refers to the pure inorganic mineral residue that remains after all the organic carbon, fats, and proteins are burned away.
Okay.
5.38% is a massive mineral payload, and the dominant mineral in that profile is potassium.
And how does a massive dose of potassium affect the kidneys?
High concentrations of potassium profoundly alter the osmotic balance in the kidneys. It acts as a powerful natural diuretic, forcing the kidneys to increase urine volume and flush the urinary tract.
Ah, it literally flushes the system.
Yes. This increased flow rate is one of the primary mechanical factors in preventing the crystallization of calcium oxalates, which form kidney stones, and aiding in the expulsion of existing small stones. The 16th century herbalists observed the physiological result without knowing the microscopic potassium mechanics.
They didn't know what a potassium ion was, but they knew the mushroom flushed the system. It is incredible trial and error. Though the historical sources do mention that the same doctors also prescribed it to cure tuberculosis, which modern science confirms was a complete failure.
It was. Tuberculosis is a profound bacterial infection of the lungs. The potassium flushing had absolutely no effect on the Mycobacterium tuberculosis pathogen. But again, their methodology was empirical observation. They threw a powerful biological agent at a disease to see if it worked. Sometimes it hit, sometimes it completely missed.
But the utility of this mushroom wasn't just restricted to ancient medicine. People actually hunt this thing for food today, which just boggles the mind. It actually has a very impressive nutritional profile. The literature notes it contains roughly 19.33 grams of protein per 100 grams of dry weight.
It's very nutrient-dense.
That is highly competitive with traditional protein sources. But how are modern foragers getting past the blistering dialdehydes? How do you make a tear gas mushroom taste good?
It is entirely dependent on rigid, traditional preparation methods that chemically neutralize the threat. In regions like Finland and Russia, where the peppery milkcap is deeply ingrained in the culinary culture and highly prized, foragers utilize a method of repeated, aggressive parboiling.
Explain the chemistry of the parboiling.
They submerge the freshly harvested mushrooms in large vats of boiling water for several minutes. The extreme thermal shock does two things. First, it instantly denatures the delicate proteins of the esterases and lipases, the enzymes responsible for triggering the chemical cascade.
So the detonator is melted before it can mix with the explosive.
Exactly. Second, the prolonged boiling physically leaches any naturally present volatile dialdehydes out of the fungal tissue and into the water. The foragers then discard that toxic water, rinse the mushrooms, and often repeat the boiling process a second or third time to ensure complete neutralization.
And then what do they do with the inert mushroom tissue?
Typically, the neutralized mushrooms are heavily salted, pickled in vinegar, or lacto-fermented over weeks. The fermentation introduces beneficial bacteria that break down the tough cellular walls, rendering the high-protein mushroom highly digestible and imparting a complex, savory flavor.
Okay, that sounds delicious, but it also sounds like a tremendous amount of manual labor just to eat a mushroom. But the deep dive sources highlight another entirely natural way to make this mushroom edible. And it might be the most bizarre sci-fi biological interaction we've covered today. You don't have to boil it for hours if you're lucky enough to find one that has been hacked by a completely different fungus.
Yes, you are referring to the Hypomyces lactifluorum interaction, commonly known as the lobster mushroom phenomenon.
Yes. Please explain the mechanics of the lobster mushroom, because it completely recontextualizes the idea of parasitism for me.
It is a remarkable event primarily observed in the forests of North America. The peppery milkcap is frequently targeted by a specialized parasitic fungus called Hypomyces lactifluorum.
Okay.
When the spores of this parasite land on a young, developing peppery milkcap, they germinate and rapidly spread across the entire exterior of the host.
Basically coating it.
Yes. It engulfs the host in a hard, dense, bright orange-red crust. Visually, the white mushroom transforms to look exactly like the cooked shell of a marine lobster, hence the name.
But the true marvel is happening on a molecular level beneath the crust.
Precisely. The parasite doesn't just sit on the outside and siphon nutrients. Its mycelium actively penetrates the deep tissues of the host mushroom and begins releasing effector proteins.
Effector proteins? What are they doing to the host?
They're literally rewriting the host's genetic expression. The parasite's proteins actively suppress the expression of the host's defense genes. It fundamentally downregulates the sesquiterpene syntheses.
The enzymes that build the chemical weapons. The parasite reaches into the host's source code and turns the weapons systems off.
Exactly. It completely halts the production of the toxic fiery dialdehydes. And simultaneously, the parasite upregulates the production of its own unique amino acids and volatile flavor compounds.
Oh.
What was once a caustic, unpalatable, tear gas-producing mushroom is biologically terraformed into a dense, highly sought-after culinary delicacy with a mild, slightly seafood-like flavor profile.
It is literally a biological hostile takeover that results in a gourmet meal. Nature is operating on a level of complexity that is almost hard to comprehend.
It really is.
And speaking of hacking biological software, this leads us perfectly into the cutting-edge modern pharmacology and synthetic biology sections of our deep dive. Because researchers today aren't just looking at this mushroom to cure kidney stones or make a nice risotto, they are extracting its chemical weapons to build entirely new drugs.
Indeed. We spent a significant amount of time detailing how aggressive and tissue-damaging those pungent dialdehydes velleral and piperdiol can be. But modern pharmacological screening has revealed that these exact same aggressive properties have incredibly potent medical applications.
Specifically regarding their antiviral and antibiofilm capabilities, right?
Yes.
Antibiofilm. Let's define that for the listener, because biofilms are a massive problem in modern hospitals.
They are a huge issue. When certain pathogenic bacteria infect the body, they don't just float around freely. They cluster together and excrete a dense, slimy polymeric matrix, a physical shield called a biofilm.
Like a bunker.
Exactly. This shield is incredibly difficult for standard antibiotics to penetrate. The bacteria essentially build a bunker, allowing chronic infections to persist.
So how do the mushroom's weapons interact with the bunker?
The 1004-dialdehydes derived from the peppery milkcap have demonstrated a remarkable ability to disrupt the structural integrity of these biofilms.
They break it down.
The reactive nature of compounds like piperdiol allows them to penetrate and break down the protective matrix, leaving the underlying bacterial colony exposed and vulnerable to standard immune responses or lower doses of conventional antibiotics.
The mushroom provides the bunker buster munition, and the notes also mention ongoing research into oncology.
Yes, there is very promising preliminary data investigating their potential anti-cancer properties. The same cytotoxic cell-destroying nature that causes chemical burns in a predator's mouth has been shown in vitro studies to exhibit targeted activity against specific malignant cell lines.
Like lung and colon cancer models.
Yes. The compounds induce apoptosis, or programmed cell death, in the rapidly dividing cancer cells.
So the same chemical mechanism that evolved to torment forest rodents could potentially be isolated to fight human lung cancer.
That is the promise of natural pharmacognosy.
But it immediately presents a massive logistical problem. I mean, if we discover that velleral is the cure for a specific cancer, how do we get enough of it? We can't deploy armies of foragers into the woods to individually squeeze spicy milk out of wild mushrooms.
No, it would be wildly inefficient, seasonal, and not scalable for global medicine.
Right. And that exact logistical bottleneck is what the 2023 to 2025 genomic mapping breakthroughs have entirely resolved. Scientists recognized that harvesting wild fungi was a dead end. Instead of trying to extract the chemical, they decided to extract the instruction manual.
They sequenced the genome.
Yes. They comprehensively sequenced the DNA of Lactifluus piperatus. And through complex transcriptomics, they successfully identified and mapped the exact gene clusters responsible for coding the sesquiterpene syntheses.
The genes that build the chemical factory.
Exactly. They isolated the fundamental genetic blueprints. And leveraging the tools of modern synthetic biology, researchers are now utilizing plasmid vectors to insert these specific mushroom gene clusters directly into the genome of a completely different organism, Saccharomyces cerevisiae.
Saccharomyces cerevisiae, which is brewer's yeast.
The exact same microscopic fungus humanity has used for millennia to ferment beer and make bread rise.
The very same. But in this context, it isn't making beer. By successfully inserting the mushroom's DNA into the yeast reads the new genetic code and begins manufacturing the mushroom's enzymes.
It's incredible.
Researchers can grow massive, industrial-scale vats of this genetically modified yeast in highly controlled, sterile bioreactors. You feed the yeast simple sugars, and instead of just producing alcohol and carbon dioxide, the yeast acts as a microscopic pharmaceutical factory, churning out huge quantities of pure velleral and piperdial.
I want you to really appreciate the magnitude of this. It is literally like taking a highly classified, complex weapons manufacturing software patch, downloading it, and successfully installing it into your standard kitchen bread maker. You pour in sugar water, and out comes a targeted, scalable, antiviral chemical weapon.
It entirely bypasses the need to ever forage in the woods again. It is a closed-loop pharmaceutical pipeline.
It is a triumph of bioengineering. It allows for the sustainable mass production of critical agricultural antefeedants and potential pharmaceutical precursors without disturbing a single forest ecosystem.
But while human scientists in sterile white coats are busy hacking the mushroom's genetics in bioreactors, wild evolution is still out there running its own raw, unfiltered experiments.
Oh, absolutely. And the very last piece of literature you brought for this deep dive, the 2026 field surveys, involves a massive fundamental evolutionary plot twist for this species.
It is a discovery that challenges our basic assumptions about evolutionary biology. Mycologists conducting deep surveys in the subtropical forests of southern China recently identified and described a brand new, very close genetic relative to the Peppery Milkcap complex. They formally named it Lactifluus ambicystidatus.
Okay, and based on everything we've learned today about how perfect and impenetrable this chemical defense system is, let me guess. The new Chinese variant has evolved an even more agonizing, even more complex chemical weapon?
No.
Wait, really?
That is the shock that rippled through the mycological community. Lactifluus ambicystidatus has completely lost the ability to produce milky latex altogether.
Wait, what? How is it a milk cap?
The lactiferous system, the intricate microscopic network of tubes that stores the inert chemicals and the enzymes, is entirely absent. The genetic pathways have been silenced or lost. It is technically a milkcap mushroom that has completely evolved away from milking.
I need to push back on the logic of evolution here. We have spent an hour detailing how flawless this binary chemical defense system is. It is instantaneous, it locks onto the TRPA1 receptor, it forces predators to remember the pain forever, it is the ultimate deterrent. Why would natural selection ever favor abandoning your most powerful weapon? It seems like an evolutionary downgrade.
To understand why it abandoned the chemical weapon, we really have to look at the harsh realities of bioenergetics. Biological systems are economies of energy. Synthesizing complex sesquiterpenes, like sterol volutinol, building and maintaining the massive network of specialized lactiferous tubes to store it safely, and constantly producing the enzymes required for the binary reaction requires an immense, continuous expenditure of metabolic energy from the fungus.
So it is incredibly expensive to maintain a massive standing army, even in peacetime.
Exactly. In the specific ecological pressures of the southern Chinese forests, maintaining that standing army apparently became too costly. Perhaps the local predators evolved immunity, or perhaps the soil lacked the nutrients to support such expensive chemistry. Whatever the pressure, that specific lineage found a cheaper, more metabolically efficient alternative.
What did it do instead?
Instead of chemical warfare, Lactifluus ambicystidatus evolved sheer physical armor.
Physical armor on a mushroom. What does that even look like? Under a powerful microscope, you can see that the external surfaces of its gills and cap are densely covered in specialized sterile cells called lamprocystidia and macrocystidia.
Break those terms down.
They are essentially microscopic, heavily thickened, rigid cell walls that protrude outward like spikes and impenetrable shields.
Wow.
Instead of burning a predator's mouth with chemistry, the mushroom relies on being physically tough, rigid, and deeply unappetizing to chew on.
So it traded the high-tech, energy-draining chemical airbag system for a permanent low-maintenance suit of microscopic spiked armor.
It is a massive evolutionary pivot. It proves that there is no such thing as a perfect or final solution in nature. There is only what works best for a specific environment with the least amount of energy spent at that exact moment in time.
It is a beautiful illustration of the dynamic, ever-shifting nature of evolutionary adaptation. Even a deeply conserved, defining trait like the chemical latex of a milk cap can be ruthlessly discarded if a more efficient passive survival strategy presents itself. Evolution is entirely unsentimental.
Absolutely unsentimental.
Okay, let's take a breath and look back at the sheer scale of what we've unpacked today. Because this has been a relentless journey. We started with the assumption of a simple, unassuming white mushroom sitting quietly on the forest floor. And from that single organism, we uncovered a global taxonomic conspiracy masking a cryptic network of genetic clones.
Right.
We explored a hyper-aggressive binary chemical weapon system that actively hunts down our tear gas pain receptors while turning off our ability to feel chili peppers.
Yes.
We learned about a manipulative or perhaps highly protective forest guardian that dictates the physical root architecture of giant oaks while hoarding toxic industrial lead in microscopic vaults. We dug into the archives of 16th century Polish herbalists using its mineral ash to flush kidney stones and saw how a parasitic lobster fungus can overwrite its DNA to make it delicious.
It's an astounding list.
And finally, we saw how modern science is literally extracting its source code to build targeted antivirals in vats of brewer's yeast, while natural evolution in China is trading that same chemical warfare for microscopic spiked armor.
When you synthesize all of those distinct fields of study, the peppery milkcap really ceases to be just a fungus. It emerges as a profound intersection of ecology, organic chemistry, human history, and the bleeding edge of synthetic bioengineering. It serves as a stark reminder that every single species sharing this planet possesses a story written in its genome that is infinitely more complex, ancient, and deeply interconnected than its simple outward appearance could ever suggest.
And that brings us right back to you. Think about your own environment. If you live near an old factory, a busy highway, or even just take a weekend hike through a local state park, think about the staggering, invisible complexity existing in the soil right beneath the tread of your boots. Humanity spends billions of dollars every year frantically looking for new synthetic drugs, searching for novel pain management solutions, and engineering massively expensive environmental cleanup technologies in sterile, white-walled laboratories. But what if the solutions to our most complex medical and ecological crises have already been perfected? What if the answers are locked away as latent billion-year-old code inside the very fungi we casually step over without a second thought? The next time you walk through the woods and your eye catches a plain white mushroom sitting in the dirt, pause for a second. Remember that you aren't just looking at the scenery. You might be looking at a biochemical masterpiece that holds the key to the future.